There's really no other tool available that we have as cell biologists other than microscopy that really show you cells in action. And it's really fascinating to me. I mean, I can spend hours, days in the microscopy room just with the cells, looking down the oculars and just seeing what they do. I'm Nihan Altan-Bonnet. I'm an investigator at the National Heart, Lung and Blood Institute. Towards the end of my postdoctoral work, I was contacted by a virologist at the National Institutes of Health who asked me if I would be willing to look at what happens to cells when they're infected with poliovirus using microscopy. So in the remaining time of my postdoc, I worked with this researcher to set up the microscopy conditions so that we could visualize poliovirus dividing, replicating inside cells. Our research has really given us a new window on viral transmission and has also opened up new therapeutic opportunities for blocking viral transmission. Imagine a cell where you have all these soldiers that have formed. So a single soldier has come in, made a replication factory, and then that replication factory has led to the production of thousands, hundreds of thousands of soldiers. And now those soldiers have to get out of that cell and go and infect new cells. And what was the sort of the paradigm in virology is that those soldiers, those viruses, they leave the cell and they go on to infect other cells as individuals, as lone soldiers. Using microscopy, what we found was completely the opposite. What we found is that viruses like poliovirus and other similar viruses, they leave their cell not as individual soldiers, but as in groups, in sacs. So these sacs, called vesicles, are shed from these cells, and they contain thousands of these viral particles within them. This was not discovered before because these sacs were being disrupted. They were being lost in the process of handling the biological materials. And whereas with microscopy, there's so little handling of the biological materials, we were able to visualize these sacs and keep them intact. Our team is really excited about the future of microscopy and its applications to virology, and we hope that this type of work that we do will lead to new discoveries and new therapeutics to tackle viral infections.